So I mentioned in the previous session that there are two main bioinformatics resources. So the first is the National Center for Biotechnology Information, NCBI, and the second is the European Bioinformatics Institute, or EBI. So let's have a look at these two resources in a bit more detail. So the NCBI is actually funded by the National Institute of Health and was initially established to store all molecular biology information. It has grown dramatically since the completion of the Human Genome Project. And what they have is that they've developed and maintained a variety of databases and resources. One of the main databases, or one of the first databases that was actually developed at the NCBI, is GenBank. And essentially, this is a database that contains an annotated collection of all publicly available DNA sequences. So if you recall, previously when I was talking about databases, this is an example of a database that is publicly accessible. So in terms of submission as well as access. So anyone is able to publicly submit sequence data to GenBank. And you're also anyone is able to download sequence information from GenBank. As I mentioned before, it is part of the ION SDC. And this database is one of the main databases that is obviously updated on a regular basis and approximately every 2 months. And there are several divisions within the GenBank database. So in terms of the divisions within GenBank, these are some of the specialized divisions or sections within the GenBank database. So we have express sequence tags, or ESTs. These are short subsequences of cDNA sequences. We then have a high throughput genomic sequence section, and these contain sequences from clone-based high throughput genomic sequence experiments. We then have a section for microbial genomes. We have a section for whole genome shotgun sequences, and we have a section for transcriptome shotgun assembly sequences. So again, the main database you will then have access to all sequences. However, if you are interested only in a subset of the database, you are then able to access information from one of these particular sections or divisions within GenBank. So this is the NCBI homepage, and what you'll find is that this homepage is usually updated on a regular basis. So things do generally change over time. But essentially, let's have a look at what the setup is like on the current version. So on the left-hand panel, you'll see that there is a list of categories or a resource list. And essentially, they've categorized all the different resources that are available at NCBI based on larger categories such as chemicals and bioassays, data and software, DNA and RNA, gene expression, literature, proteins, etc. So if you click on any one of those resources, it will open up a list of all the resources that are available in that particular category. In the center panel, what you'll find is a number of different links to different parts of the NCBI website. So you'll see the first two are submit and download. So if you click on these, this will take you to pages. So if you click on the submit link, it will take you to a page that will give you information and step-by-step information on how to submit sequence data to NCBI. So again, if you're doing any project whereby you're generating sequence data, in most instances when you publish, you would want to submit that data to one of the main primary database repositories. You then can also download data directly to your computer. And again, there's different ways that you're able to do that, and you can click on that link to find out more about how to download information from NCBI or any one of the resources at NCBI. They also have a number of tutorials on how to access and to use the NCBI resources. Then there's a link for development. So these are basically small programs that have been developed in order to access their data directly from the back-end databases of NCBI. You then have Analyze, which will take you to a list of tools categorized according to different types of data analysis. And then there's a Research tab, which will take you to a page documenting some of the research projects that the NCBI is involved in. On the right-hand side, which is usually quite useful, is they have a list of the popular resources that are used at NCBI. So mostly, if you're using some of the more popular resources, that's a quick way to get to some of the popular resources that are based at the NCBI. So what I've done here is I've clicked on the Analyze link, and what you'll see is it'll take you to a page with a number of tabs. I'm currently on this page. I've clicked on the Genomes tab, and essentially it'll give you a list of tools that are available for doing some type of analysis based on genomic sequence data. Okay, so for example, there's a 1000 Genomes browser, which will allow you to access 1000 genomes data, and we will talk a bit more about 1000 genomes data later on in this course. There's a link to BLAST, which allows you to search against the databases at NCBI using a sequence of interest. And again, we'll have a look at BLAST later on in this course as well. So on the top list of panels, you'll see that there's all tools, and then there are particular tabs related to the different types of or different categories. So for example, tools for searching literature, tools for dealing with finding health or clinical information, tools for dealing with genomes, tools for dealing with genes, proteins, chemicals, etc. So as I mentioned before, there's also the Learn tab on the main homepage, and that gives you a link to a list of tutorials. And this is pretty good to use if you are using NCBI for the first time. So what the NCBI does is they have a number of online sort of courses that you can do, or tutorials and webinars that they presented at institutes, and it's available for you to then just view these courses and tutorials in order for you to become familiar with the tools that are available. They also have extensive documentation for all their tools and resources, which I suggest you do have a look at if you are going to be using one or more of the tools at NCBI. So what I've done now is I've clicked, remember on the left-hand panel, there were these different categories. And what I've done is I've clicked on the DNA and RNA category. And what you'll see again is what the web page will present to you is a list of all the tools, databases, and ways of analyzing and accessing DNA and RNA data. Okay. So on the first tab, it'll have, the first tab is all the resources. The second tab is databases. So these are the particular databases at NCBI that store DNA and RNA information. The third tab is download. So how would you then be able to download DNA and RNA data? The next tab is submission. So again, how would you submit data? Then there's tools, and then there's the how-to tab. So if you click on each one of these, it'll give you the different resources in that particular category. So another thing to keep in mind is that there's not only DNA data stored at the NCBI, and there aren't only tools available for analyzing DNA data, but there's a number of different categories for doing various different types of analyses. So I do encourage you to then explore these if you're not dealing with DNA data, because there might be some resources that are applicable to the research area that you're looking at. So in the next session, we'll have a look at the EBI or EMBL resources in more detail.